Hello, everyone. This is Group 5, and we will be discussing cell death. Cells can undergo cell death to regulate development and maintenance of tissue homeostasis. Two forms of cell death can occur. Apoptosis is one form, and necrosis is the other. Apoptosis is a programmed control process that requires energy. Examples are the separation of fingers and toes. It promotes morphological changes that minimize the impact on neighboring cells to avoid promoting an immune response. Apoptosis causes the cell to shrink and become condensed. The cytoskeleton will break down, the nuclear envelope will dismantle, and the DNA will become fragments. Blebs are formed to avoid release of cellular components into the extracellular matrix. The membrane's phospholipid content is altered, which makes the dying cell more susceptible to phagocytic attack and removal. Necrosis is an irreversible premature sudden cell death due to external factors. It is an uncontrolled passive process. An example would be skin being exposed to poison or a heart attack. Necrosis can also be the death of living cells. It undergoes a swelling process that allows cytoplasmic vacuoles and blebs to be made. The ER enlarges and ribosomes are then disaggregated. Once this occurs, the elements are released and the nearby cells, as well as the damaged ones, have inflammatory reactions due to lack of blood. In relation to cancer, apoptosis plays a vital role in the immune system. Cancer can be derived from the lack of apoptosis, while an excess of apoptosis can lead to loss of cells. The failure of a cell to produce apoptosis due to a mutation or infection can begin to replicate uncontrollably, which will result in tumors.
Apoptotic signaling occurs in two different ways: by the extrinsic pathway or the intrinsic pathway. Extrinsic pathways are usually activated by the immune system. These signals come from outside of the cell, and the ligand from the signal binds to the receptor of the cell, which will eventually lead to death of that particular cell. Intrinsic pathways are activated by internal damage, like damaged DNA, for example. The intrinsic pathway is then activated by the release of cytochrome C from the mitochondria that leads to the death of the cell. Growth factors are usually a protein or steroid hormone that can stimulate or inhibit division. Two types of growth factors are platelet-derived growth factor and epidermal growth factor. Platelet-derived growth factor is a protein that stimulates the development of connective tissue cells and smooth muscle cells. Epidermal growth factor is distributed in many tissues and body fluids. Growth factors such as these bind to receptors on the surface of target cells, which leads to kinase activity, leading up to the activation of RAS pathway. The pathway shown here leads to the activation of protein kinase AKT. Growth factors that bind to receptor tyrosine kinases activate this pathway. AKT suppresses apoptosis by phosphorylating and inactivating a protein that normally promotes apoptosis called BAD. AKT also prevents cell cycle arrest by activating TOR, leading to cell survival.
I'm going to talk about the major steps in apoptosis. When it comes to apoptosis, internal contents of the cell are dismantled. In the beginning, chromosomes condense and the cytoplasm becomes smaller. The cell then begins to produce something called blebs, which are cytoplasmic extensions. The nucleus begins to fall apart along with the organelles, and the DNA of the cell is pinched by DNase, which is an enzyme that digests DNA. Eventually, the cell is all over the place in small pieces called apoptotic bodies. These apoptotic bodies are then eaten by phagocytic cells. How is apoptosis triggered? One well-known cell death signal is CD95. CD95 is a protein on the surface of infected cells in the human body. When cells are infected by certain viruses, cytotoxic T lymphocytes are activated and induces the infected cells to initiate apoptosis. Also, apoptosis can be triggered indirectly by the removal of survival factors, and the major site that plays a role in this is the mitochondria. Mitochondria's role. So how do mitochondria play a role in cell death? Mitochondria trigger apoptosis by releasing cytochrome C into the cytosol. This happens when a bunch of pro-apoptotic proteins pile up at the surface of the mitochondria and makes little pathways for cytochrome C to escape into the cytosol. Too much cytochrome C in the cytosol leads to apoptosis in the cell. Another way mitochondria is involved is when a cell is so damaged it cannot repair itself. So when this happens, the cell undergoes apoptosis by the activity of p53. p53 binds to a protein called PUMA and inhibits BC-2. So basically, the p53 pathway eventually activates pro-apoptotic proteins to induce apoptosis within the cell.
Caspases plays a major event that occurs in apoptosis by the activation of a series of enzymes. Caspases are known to be a central effector of apoptosis. Caspases can be defined as a family of protein enzymes that play central roles in programmed cell death or inflammation. Caspases are produced as inactive precursors. These precursors are known as procaspases. Procaspases can be defined, they cleave or to create active enzymes, in most cases by other caspases in the proteolytic cascade. Once they are activated, caspases cleave other proteins. Caspases can be divided into different functional groups: effectors and initiators. Effector caspases manage the regulation of apoptosis, and initiator caspases initiate signals within apoptosis. Effectors complete the mass proteolysis that leads to the promotion of apoptosis effectors. Effectors are created in the cell as dimers and proteolytic handling by an initiator compound which is required to trigger their action. On the other hand, initiator caspases are translated as monomered zymogens. Caspases can be classified as initiators and as effectors. For initiator caspases, I'm going to be talking about caspase 8, and for effectors, caspase 3. Caspase 8 is strictly only detected in gastric cancer. The rate of the mutation in gastric cancer is higher than those of breast cancer and leukemia, just to be a few. Caspase 3 is activated by both intrinsic and extrinsic cell death pathways. It plays an important role in the execution period of cell apoptosis. The mutation that is within caspase 3 has been accounted for in the MCF7 breast cancer cell line
proposing the existence of caspase-3 mutation in human tumor tissues. So, this figure is showing what exactly happens inside the cell when it receives a cell death signal. So first, the cell receives a signal that tells it to die. In this diagram, it's the ligand on the surface of a cytotoxic T lymphocyte that binds to the death receptor of the target cell. The binding causes receptors to bunch together and adapter proteins to come to the site, resulting in clustering of initiator procaspases proteins that now become activated in step 3. The procaspases now activate the executioner caspase, called caspase-3. When survivor factors are no longer present, death-promoting proteins accumulate, outweighing proteins that are anti-apoptotic, causing a release of cytochrome C. Cytochrome C forms a complex with other proteins, leading to activation of caspase-9, which is an initiator caspase. Caspase-9 then activates caspase-3, which is the executioner caspase, causing apoptosis. So, BCL family of proteins. The BCL-2 family of proteins is known as an important gatekeeper to the apoptotic response. This group of proteins compromises pro-apoptotic and anti-apoptotic members that interact with each other. The BCL-2 family can be classified into 3 functional groups: anti-apoptotic proteins, pro-apoptotic effectors, and pro-apoptotic activators. Activators are important mediators in the cellular response to stresses such as DNA damage. Effectors are those BCL-2 proteins closely associated with the mitochondrial membrane, and when stimulated by the BH3-only activators, they promote the pores in the mitochondrial membrane, starting apoptosis. Signaling. In healthy cells in the outer mitochondria, there are anti-apoptotic proteins that block cell death by apoptosis only if the cells are constantly exposed to survival factors. In contrast, pro-apoptotic members trigger the release of caspases from death antagonists by inducing the release of mitochondrial apoptogenic factors into the cytoplasm. The reasoning behind why different cells have a huge range of capabilities and morphological features is because of gene expression, starting with the nucleus at the genomic level all the way up until after translation. This activation and deactivation of genes is what determines what type of protein is produced, when it's produced, and how much of this protein is produced.